Okay, so this is time. I would like to let Xinyi. Can you unmute yourself to introduce today's speaker?
Sure.
Okay, go ahead.
So it's my great pleasure to introduce our first speaker of this semester, Dr. Teresa Bergholz. So Dr. Teresa Bergholz is an associate professor in Department of Food Science and Human Nutrition at Michigan State University. She earned her PhD in food science from MSU in 2007, and then completed her postdoctoral research training in the Department of Food Science at Cornell University. Her research program focuses on understanding pathogen survival in food systems, as well as developing effective control measures. So without further delay, let's welcome Dr. Bergholz, and I will pass it to you.
Wonderful. Thank you so much for that introduction. I really appreciate the invitation to talk with you everyone today and provide an opportunity for me to share my group's work over the past few years as we assess different methods for controlling foodborne pathogens on wheat grain. And so I want to start off with a question. And so maybe you guys can raise your hands if you've seen some of the signs like this, right? Like the one at the top that says life is short like the spoon. How many people have seen messages or signs like this before? Anybody? No? Okay, well, how many people have eaten raw cookie dough before? All right, we've got a few hands raised here. And so what I'm going to talk about today is the concerns with consumption of raw flour and some of the research we've been doing to reduce STEC and salmonella on wheat grain. And so the issue of sugar toxin producing E. coli present in flour became national news after this multi-state outbreak in 2016. This was a widespread outbreak over 24 states, and 63 people were infected and 17 people hospitalized. And when they talked to and interviewed the patients from this outbreak, they found that 50% of them that were ill reported eating raw dough. So this led to a massive recall of flour, about $20 million of recalled product, and was one of the major outbreaks that really put this problem in everybody's radar. And so since 2016, there have been 3 more outbreaks of STEC that have been linked to flour and flour-containing products in North America. So in 2017, there was an outbreak in Canada. 2019, there was another STEC outbreak in the US. And then most recently in 2021, there was an outbreak linked to cake mix with 16 illnesses over 12 states. And since 2009, there have been 18 recalls of flour products for STEC contamination. And so this has led to a number of outreach messages like the screenshot from Twitter that I captured here from USDA, pointing out that raw dough and batter are never safe to eat, even if they don't contain raw eggs, really highlighting the issue of pathogens in flour. So in addition to sugar toxin producing E. coli, we've also seen outbreaks of salmonella that are linked to flour. In 2005, there was an outbreak that was linked to cake mix. And in this case, the contaminated cake mix was used to make cake batter flavored ice cream. More recently, there was another outbreak linked to cake mix in 2018, and there have been at least 4 major recalls of wheat flour-based products since 2009. So after these outbreaks, there were messages added to bags of flour, such as this example photo here at the top of the screen that says cook before sneaking a taste. And researchers were interested to understand more about consumer behavior and how they use raw flour at home. And so the data that I have here summarized on this slide was from a paper published in 2020 from Betty Fang's lab at Purdue. And so they found that 2/3 of respondents reported consuming raw cookie dough or batter, even though 40 to 50% of them were aware that this could be a food safety risk, although most people thought that the risk was due to consumption of raw eggs that were in the batter or the dough. And I think one of the most interesting things from this research was that 85% of people had never heard of any recalls related to flour or cake mix. And so while Flour is not a ready-to-eat food. This data shows that there are a number of consumers that are treating it as such. And so I just want to give a little bit more background on the pathogens of concern that I'm going to be focused on for the rest of my talk. So Shiga toxin producing E. coli, also referred to as STEC or sometimes as enterohemorrhagic E. coli or EHEC. These are characterized by their production of Shiga toxin, which is the virulence factor that leads to hemorrhagic colitis and complications such as hemolytic uremic syndrome. This pathogen is estimated to cause about 265,000 illnesses and 30 deaths each year in the US. The main serotypes that you're likely to be familiar with are things like O157H7. This has been associated with a number of outbreaks linked to products like ground beef, fresh produce, and then some of the other serotypes such as O26 and O121, which were associated with these flour outbreaks that I just talked about. For Salmonella, these are responsible for over 1 million infections each year in the US. The main symptoms are typically gastroenteritis, but can lead to systemic infections as well. Additionally, we have the concern for multi-drug resistant Salmonella infections. Some of the Salmonella serovars that have been reported as associated with low moisture food outbreaks, including flour, are those such as Typhimurium, Agona, Tennessee, Igbeni, and Monteverde. And so when we think about how these pathogens could be getting into flour, remember both of these are bacteria that have zoonotic reservoirs. You can find them in gastrointestinal tracts of cattle, chickens, and this can then spread the pathogens into the food production environment through feces. We still don't have really great information on how exactly wheat grain can be contaminated, but some of the likely sources of contamination include through irrigation water. There's been one study that showed that STEC 0157 could contaminate wheat plants if it was present in irrigation water. There can also be a contamination present in the soil, can be deposited by birds or rodents, and then weather changes, especially when there's increased rain and flooding events near the time of harvest, that can also lead to increased presence of pathogens. During transportation of grain, there's the potential for contamination through cross-contamination as well. And so once harvested, the grain can be stored for months before further processing. The processing steps of turning grain into flour include first a cleaning step, and this helps remove any field debris, separate that from the wheat grain. And then it goes into tempering. And this is where we're adding a small amount of water to the grain so that it's absorbed by the bran layer, helps toughen that bran layer, and then makes it easier to remove in a large piece rather than being removed in smaller pieces. And then the endosperm that remains once the bran is removed is what's milled into flour. And so the class of wheat determines the type of products that it's used for. So hard wheat, this has a higher protein content, so it means this has more gluten present. And this is typically used to make bread flour. We need that nice gluten structure to help make our nice tall bread. And then soft wheat has a lower protein content, and this is typically used to make cakes and cookies. And so there isn't much information on the presence of pathogens in wheat grain, but the most recent information was published in 2019. And here they surveyed wheat grain and they sampled it from rail cars as those cars were entering mills. And this was done over a 3-year period at different locations around the US. And they tested almost 4,000 samples, and they found, so the data that's circled here at the bottom in total, so they found that there were less than 0.5% of samples were positive for enterohemorrhagic E. coli, and a little over 1% of samples were positive for Salmonella. So there's not a very high prevalence of these pathogens on the grain, but one of the things that this pointed out is that it is likely that the grain can be contaminated before it enters the mill, and that can be an issue. And so this leads us to the research questions my group has been working on. So first one is just to understand more about how long pathogens can survive on grain. There have been a number of studies published looking at how long the pathogens can survive in flour, but had previously not looked at survival on the grain itself. And since we know that grain can be stored for many months from harvest to when it's actually milled, this was important to understand the dynamics of pathogen survival. And we also wanted to determine what types of processes can be used to inactivate pathogens on grain. An important consideration here is to be sure that we're maintaining that flour functionality. So I just mentioned the different end uses of different types of flour. And so especially for bread flour, we want to maintain that gluten functionality, which can limit the types of treatments we use to inactivate pathogens. Okay, so this first study I want to share with you was conducted by Jess Lauer. She was a master's student in my group when my lab was at North Dakota State University, and she wanted to know how long Aztec and Salmonella can survive on wheat grain. And so based on studies in other low moisture foods, such as almonds, we know that pathogens can survive for months or even years on this type of food. And so she wanted to conduct this study over the course of a year. So she inoculated hard wheat kernels with Salmonella and Aztec strains. So there were 10 different strains that she selected, and they were all evaluated in triplicate. She inoculated the grain for a final density of 6 log CFU per gram. And the method she used for inoculation was to grow the pathogens on agar plates as a lawn, and then scrape the lawn off of the plate and use a minimal amount of water when it's being mixed with the grain. So that way we're not increasing the water activity by a significant amount. It does still increase. And so the inoculated grain, we then equilibrated that back to its original water activity of 0.52. And then these were portioned into Mylar bags and stored at room temperature, so 20 to 22 degrees Celsius over the course of the year. And so she set up the experiment and then just looked at survival at different points over this year. And so what we're looking at here is the log CFU per gram, which is on the y-axis, and by over time, in this case, the time is represented in weeks. And you can see that there's 2 clusters of data here. The first one, the top set are 4 Salmonella strains, which had much greater survival compared to the Aztec strains, which are the group that's shown here kind of in the bottom. And there were some of the Aztec strains that reached the limit of detection around 40 to 44 weeks of incubation, whereas the Salmonella strains, we were able to detect them throughout the entire year of the study. So we saw that Salmonella had a greater survival. She then calculated the time it takes for 1 log reduction for each strain. And she found that this was about 22 to 23 weeks for these Salmonella strains. But it was only about 12 weeks for a 1 log reduction of our Aztec strains. So while both of them survived for fairly lengthy periods of time, the Salmonella strains had a slower rate of decline compared to these Aztec strains. And so this data showed us that the pathogens are capable of surviving on the grain. And so the next step, what we wanted to look at was what happens to the pathogens if they're present on the grain during the tempering step. So during tempering, I mentioned earlier, we're adding water to the grain to toughen that bran, making it easier to remove during milling. There's a little bit of data published in the early 2000s that showed that when they looked at just total plate counts, so aerobic bacteria and coliforms on grain, they found that there was a slight increase in those numbers from comparing before and after tempering. And so we wanted to know how the addition of water during tempering impacted pathogens that had been purposely inoculated onto that wheat grain. And so this work I'm going to share with you was done by Yao-Wei Lin, who was a master's student in my group at MSU. She actually just defended her master's thesis last week. And so she used a similar set of strains that Jess did for that storage study, and the same inoculation process, right? Where we're growing the strains as a lawn and then scraping that off and using that to inoculate our different batches of grain. And again, the water activity was equilibrated to 0.5 before we assessed what happens during tempering. And so for this experiment, we used the AACC method for lab scale tempering of grain. So this is the standard process that's used for evaluating tempering and milling of grain when we're testing it for different quality parameters. And in this case, we're using it for evaluating how our bacteria change during the process on the inoculated grain. So the first step of this is to measure the initial moisture content of the grain, and then use that information to determine how much water should be added to the grain to achieve this 14.5% moisture content. Once that water is added, so in this case, we're using 50-gram samples, and they are in like 250 ml bottles. Once the water is added, they're placed in a rotational device so that will essentially tumble the water and the grain together in their containers for 30 minutes. And after that, you just let them sit at room temperature for 15 hours. Measure the moisture content again. And so our final target moisture content is 15%. So if any water needs to be added, that final amount of water is added. And then they're rotated again for 30 minutes. And so what Yahweh did was she measured bacterial numbers at the following time points that are listed here under the enumeration time part of the slide. So at the very beginning, before any water is added, half an hour after that water is added, and then at time intervals through 16 hours. And it's important to note here, so we're talking about adding water. The total amount of water that was added to these samples is about 3.5 milliliters of water when we have a 50-gram sample. So it's still a fairly small amount of water. The idea is that you just have enough water to coat the surface of the grain so that can be absorbed into the bran layer. And so what we saw that even this addition of a fairly small amount of water has a big impact on the water activity of the grain. So what we're looking at here is the water activity and how that changed over time for our wheat grain samples that were inoculated with salmonella with STEC, and then those that we left uninoculated to quantify what happens to the native aerobic microbes on the grain. And so what you can see here is that the water activity increased to about 0.85 in the first half hour after that water is added. And then there's also another increase with the second addition of water at the very end, right, between 15.5 and 16 hours. So we see that the water activity does increase. And so we also wanted to know what happened to those pathogen numbers. And what we saw, there's actually fairly minimal change in pathogen numbers on wheat during tempering. So the plot we're looking at here is the distribution of changes in cell density. In this case, we're looking at what happens over that first half hour with the log change on the y-axis, and then our aerobic microbes, salmonella, and STEC represented as our different box plots here. And so there's very little change. None of these were significantly different from zero. And then when we take a look at what happened over the entire course of the experiment, so the full 16 hours, again, we see that there's fairly little change. And again, these were not significantly different from zero. So we don't really see any evidence for pathogen numbers increasing significantly when this water is added during tempering. The next thing Yahweh wanted to know is if the cells actually can migrate around the wheat kernel during the tempering process. And so when we had initially spoken to some people in the milling industry, when we started this project, there was some interest in knowing if pathogens would migrate to the crease of the wheat kernel. And so if you look at the diagram that's shown here, there's a cross section of the wheat, and there is a crease on the seed here, and that could make it more difficult to remove microbes if they're present in that particular location of the grain. And so we wanted to know if the water provided during tempering could facilitate movement of the inoculated cells into the crease of the grain. And so to evaluate this, we took one strain of salmonella and one strain of the STEC 0121, and tagged it with M. cherry, so it's expressing this red fluorescent protein. And then used the 10x objective on confocal scanning microscope to collect the images of where the cells are on the surface of the kernel of wheat. And so what we're looking at here in the panel A, so there are arrows pointing to where the brighter spots are, but there was a fair amount of fluorescence from the wheat kernel itself. And so after we applied background threshold correction, you can then see that there still are small red spots there indicated by the arrows, which are the salmonella that was inoculated onto this particular wheat kernel. And so with this information, she then divided each kernel into different sections. So that with the crease side of the kernel looking at the left, the right, and then the crease itself. And then on the reverse side of the kernel, using that as one whole area. And so she took wheat grain inoculated with both of our strains, the Salmonella and the Aztec L121, went through the tempering process and then compared the areas at which you could observe cells on the kernels before and after tempering. She did this for randomly selected kernels over 3 biological replicates of each of the strains. And so what she found was that there was actually no difference in location of the pathogen cells on the kernels before and after tempering. So the plot that we're looking at here, so the y-axis is the percent of fluorescent signal that's above that threshold. And as you can see, most of the numbers here are fairly low. So we don't have a lot of cells inoculated on each of the kernels. And then the sample, so the symbols that are here as part of these box plots, so the triangle is when they were sampled before tempering, and the X is when they were sampled after tempering. And so there are 2 panels here for the Salmonella and for the Aztec. And she didn't find any significant differences in the fluorescent signal either across the kernel areas or among the 2 different pathogens. So based on this experiment and how she set it up, we don't see any evidence for pathogens migrating to the kernel, or sorry, to the crease of the kernel during the tempering. And so just to summarize what we've talked about so far, so we first looked at how pathogens can survive on grain, and we found that Salmonella and Aztec could survive when it's inoculated on grain and stored at room temperature, anywhere from 40 to 52 weeks. We did observe a higher rate of inactivation for those Aztec strains. We did see that water activity increases during the tempering process, but does not seem to facilitate pathogen growth. We saw minimal changes in the pathogen numbers during that tempering process. And we also did not find any evidence for migrations of pathogens to the crease of the kernel during tempering. And so what we can see so far is that if the pathogen is present on the grain, it's likely to hang around there. Right? So we know that it can survive for a fairly lengthy period of time, and it doesn't really seem to move during that tempering process. And so this leads us to the next section, which is talking about potential control measures, right? So we know that pathogens can be there, they can survive for a long time. What can we do to try to inactivate them? So there are 2 different things I'd like to talk about. The first one is addition of different chemicals during the tempering process that could potentially inactivate pathogens on the grain, and then also looking at thermal treatments specifically for the grain. And so tempering treatment, so this is been evaluated by a number of different people. And the idea here is that we're just adding some sort of antimicrobial to the tempering water. Been 2 different studies that have reported looking at different concentrations of lactic acid to the tempering water and how that impacts Salmonella and Aztec. So we've seen anywhere from a 1.75 to 2.6 log reduction of Salmonella and Aztec when lactic acid is included. In one case, they're looking at lactic acid as well as a very high concentration of sodium chloride. Another study also looked at 5% sodium bisulfate. We also saw about 2.5 log reduction of Aztec. So it does seem like there's some promise here for adding different antimicrobials to the tempering water. And so the study that we did, this was also done by Yahweh. And what we realized was that chlorine solutions, so anywhere from 400 to 700 parts per million chlorine, are already being used in some mills for mold control. In this case, they're adding the chlorinated water during tempering to control mold growth in the tempering bins. So for this study, we focused on using hard red spring wheat. We actually Yahweh created some rifampicin resistant strains. So she's selected for rifampicin resistance in the strains that we're inoculating onto the grain. This just made it a lot easier to quantify those pathogens in the presence of the background microbes on the wheat grain. And for this study, she actually inoculated the grain following the same process that I've described. And she stored the inoculated grain for 1 month after the water activity had been equilibrated. And this was to mimic the situation that we know pathogens are most likely to be associated with the grain for some time before they're processed. And then the chlorinated water solution was just made very simply by dissolving calcium hypochlorite in water and then adjusting the pH to 6.5 with a citric acid solution. And so she used 3 different levels of chlorinated water, which are shown here as part of this graph. So 400, 800, and 1200 parts per million chlorine were used, and these are represented by the different colors in the box plots here. So the green is 400, 800 is yellow, and 1200 is purple. And then each of the panels that you're looking at here corresponds to one of the different strains that was tested or the native aerobes that were found on the wheat grain. And what's being shown here on the y-axis is the log reduction in numbers of that particular strain at each of those chlorine concentrations. And so you can see here, for the native microbes, there's actually fairly little reduction of those due to the chlorinated water. For Salmonella PT30 and the Salmonella agona strains, we see about a 2 log reduction for both of these strains, and this occurred no matter the concentration of chlorine that was used. So that's why it's indicating here not significant. That's because there was no significant difference in the reduction depending on the concentration of chlorine. For the Aztec 0157 strain, you see that this was also fairly small reduction here compared to some of the other Aztec strains, which were about a 1 log to 1.5 log reduction due to addition of that chlorine in the tempering water. And so we can summarize this change in the microbial loads of Salmonella and Aztec. So in this case, we're looking at the strains of Salmonella and the strains of Aztec all in aggregate. And you can see at the 400, 800, and 1200 parts per million concentrations, in all of those cases, we see greater reduction of Salmonella, so about 2 log reduction compared to the reduction of the Aztec strains. So in addition to looking at impact on inactivation of these different pathogens, we also wanted to look at the impact of using the chlorinated water on bread baking and chloride concentration in the resulting flour. And so these data were collected by our collaborator, Dr. Shimei Shimshek, and she's currently the chair of the Food Science Department at Purdue. And so after the chlorinated, so this was done with our uninoculated wheat. It was tempered with the chlorinated water at our different concentrations here. And then they milled the grain into flour and did some bread baking tests with this. And so what we're looking at here in this table are some of the characteristics you would use to measure bread quality. So the fermentation height, the oven rise, low volume, specific volume of the bread, and then the texture and the crumb color. And so for all of these metrics, we found that there was no significant difference whether the grain had been tempered with just plain water or any of the concentrations of chlorine in the water. And so that's a good sign showing that if we want to propose this type of treatment to reduce pathogen numbers, it also does not have an impact on end use quality for these types of products. And for this study, we just focused on bread baking because the wheat grain that we had used was the hard wheat, which is typically just used for baking bread. The other quality parameter they looked at was the chloride level in wheat and then the milled products from that wheat. And so again, we're looking at the treatment levels of chlorine and the chloride that was found in the bran, the shorts, and the flour. So the shorts is like a mixture of germ that's removed when the bran is removed as well. And so again, we see here that there's no significant difference in the amount of chloride found in the flour, whether that grain had been treated with no chlorine or the highest concentration of chlorine. So that's also a really good sign that the chlorine that's applied to the surface of the grain isn't being absorbed into the flour and carried, or absorbed into the endosperm and carried into the flour. So we also then wanted to compare our data with some of these other temperate treatments, right? I gave you some examples of lactic acid, for example, at the beginning. But one of the things we realized was that direct comparison can be fairly difficult because different inoculation methods have been used. The one that used a very similar inoculation method to what we used was a paper that was just published and just came out last week from Dr. Linda Harris's lab. And they looked at differences between tempering with lactic acid when they allowed the pathogen to dry onto the surface of the kernel or when they didn't. So they just inoculated the pathogen onto the kernels and then went straight into the tempering process. And they found that there was a greater log reduction without drying. So allowing the pathogen to dry on the surface of the kernel would definitely be more of the worst case scenario where you're allowing that to adapt to that desiccated condition. There's also differences in other studies where they've grown the cells in liquid culture, and we know that that can impact desiccation tolerance, which could also then impact your results that you see during the tempering treatment. So the most direct comparison we could make, so with our chlorine tempering, we saw about a 1 to 2 to a 2.1 log reduction for salmonella in most STEC, with the exception being that one STEC 157 strain. And this was in line with the 10.9% lactic acid tempering that was reported from John Harris, where they saw about a 2 log reduction of STEC under those same conditions. And so some of our ongoing work on tempering treatments. So one of the things that we're interested in working on right now is looking at an inoculation process that incorporates a few different features. So one of them is being sure that we're allowing for desiccation of pathogens onto the surface of the grain, because we think this is the most realistic condition, the most realistic way that the pathogens would be present on the grain. And then also looking at storing the inoculated grain for some common period of time prior to tempering to also allow for that desiccation tolerance and any adaptation that would occur during that time. And then we'll also like to evaluate tempering treatments from the same batch of inoculated grain. So this would allow direct comparison of efficacy across different treatments. So we'll do side-by-side comparisons of some of these different tempering treatments on the same batch of inoculated grain. And we also want to be sure we're mimicking those industrial processes. So remember when I started talking about the tempering process at the beginning, I said we're following that AACC method. And that is designed to be standardized across different labs when they're evaluating those flour quality properties. But the way that tempering occurs in the milling industry is different than what is used by ACC. So we know that the tempering time can vary for different classes of wheat, and the target moisture also varies for different classes of wheat. So we need to incorporate some of those features into our future studies as well. Okay, and so then we'll move on to our second part here of treatments that we can use on grain. And one of these is thermal treatments, right? So we know that most thermal treatments, one of some of the benefits are that we can have a very effective 4 to 5 log reduction. But some of the drawbacks are this can be a high cost or fairly significant cost. And we know, as I mentioned earlier, specifically with flour, we have to find the balance of the treatment that will allow for effective pathogen reduction, but also maintain that flour function. And so one of the things my group had been working on for a few years is using vacuum steam pasteurization for different seeds and grains. And the idea here is that if we're reducing the atmosphere, so providing a vacuum environment, we can maintain steam, so saturated steam, at a lower temperature. And so for those foods that may be more sensitive to higher temperatures during processing, this is a way in which we can provide steam on the surface, which helps facilitate transfer of heat and higher rates of killing of pathogens, but that this can occur at temperatures less than 100 degrees Celsius. And so this work was started by Manoj Shah, who was a PhD student in my lab when I was at North Dakota State. And he worked in collaboration with a company that had a pilot scale facility in Fargo, which is called Napisol. And they worked together and inoculated Salmonella and E. coli O57H7 on a variety of food products, including flaxseed, peppercorns, quinoa, and sunflower kernels, and found that for the most part, could achieve a 5 log reduction for some of these products at 75 degrees Celsius, but definitely at 85 degrees Celsius. Our collaborators with Napisol, they actually moved their facility. And so after that, we developed a lab scale vacuum steam pasteurization system that had a capacity of about 1 kilogram. And then we used this for further studies with wheat grain. So we had done these original studies on sunflower kernels and flax seeds prior to that big outbreak of 2016. And then once that outbreak happened, we started working on wheat grain at that point. And so all of this work was done in collaboration with Dr. Shania Shimsek, in this case with her graduate student, Jane Snelling. And here, we looked at using this vacuum steam pasteurization process on hard wheat kernels, and we were trying to figure out what was an optimal temperature to treat the grain and also maintain flour functionality. And so you can see here from my picture, these are bread baking results. And so here's our control. And then this is from grain that had been treated for 8 minutes at the target temperature of 65, 75, and 85 degrees Celsius. And we could see that there was a pretty big decrease in quality, even when the grain was treated at 75 degrees Celsius. So we focused on 65 degrees as our pathogen inactivation temperature because we didn't see any significant differences in the bread quality. And when we did this and inoculated the wheat grain with Salmonella and E. coli ON21, we found that we could get about a 3 log reduction of E. coli, about a 2.5 log reduction of Salmonella during our treatment time. But the D-value for Salmonella was fairly high under these conditions. And after we did this study, we decided we would focus on soft wheat, and we hypothesized we could probably treat this to a higher temperature because the gluten functionality is not as important for cakes and cookies, which is mainly what soft wheat flour is used to make. And so we did the same type of experiment where we, in this case, looked at soft wheat kernels and pasteurized them at 65, 75, and 85 degrees Celsius, and then did baking quality evaluations. So here, what we're looking at in this part of the figure are sponge cakes that were made with the flour that had been pasteurized. And there was no difference in the cake quality parameters at any of the temperatures. This panel right here, we're looking at cookie quality. And again, we don't see any significant differences at any of the pasteurization temperatures. They also did a bread baking study, and you could see that the grain that had been treated at 85 degrees Celsius, this was definitely an inferior loaf of bread here. And so we focused on pathogen inactivation at 75 degrees Celsius because we still had good quality bread at that point. And here we can see that we could get a 5-log reduction of both pathogens and D values for about 3.5 minutes for each of them. And so this was really promising study. We thought this was something we could definitely work on improving our process of vacuum steam pasteurization. And so when my lab moved to Michigan State, we collaborated with Teresa, who was in Dr. Brad Marks's lab, and he helped us design a new updated vacuum steam pasteurization unit that had a larger capacity, so we could now treat 2 kilograms of wheat, and allowed us to have better control over temperature. There was an insulated container that was used, and we had a PLC that controlled entry of steam into the unit and recorded temperature constantly. And so in this case, we're evaluating this new unit. We focused on, our previous studies, we'd only done 1 strain of Salmonella and 1 strain of S-Tech. Here we expanded our strain set to include multiple strains of Salmonella and S-Tech. And we also included Enteric, which is a common strain that's used as a surrogate in the moisture-free Bergholz studies. So when we move to the next phase, which would be to try to validate this at a larger scale, you don't want to introduce pathogens at that point. And so knowing which strain is an effective surrogate is important. And so for this study, we again use that same inoculation method. In this case, we're targeting 8 log CFU per gram for each of these strains, so we can be sure that we can capture a 5-log reduction if that occurred. And so one of the things that we noticed here is that we had 2 portions of our thermal treatment. So what we're looking at here is the temperature in Celsius and time in seconds. And this is an actual thermal profile from one of the trial runs in the vacuum steam pasteurization unit. So the samples are first preheated to 45 degrees Celsius. So in this 2 kilograms of filler wheat, and then our inoculated samples are placed in bags inside that 2 kilograms of wheat. And then you can see that there's this fairly lengthy come-up time as the product is heated from 45 degrees Celsius to the target temperature of 75 degrees Celsius. And so that's when it reached 75 degrees Celsius, that's when we started our treatment time, right? And so this would be the relatively isothermal part of the treatment. But as you may guess, can be a fair bit of reduction in bacterial numbers that occurs as it's heating up from 45 to 75 degrees Celsius. And when Yahweh looked at this, she found that the come-up time here could range from 13 to 22 minutes. And the reduction during that time could be anywhere from 0.75 to 3 log CFU per gram. And so we knew we needed a way to include both the time and the temperature history in trying to evaluate what the reduction values were for this treatment. And so we went down the hallway to Dr. Kirk Dolan, who has done a lot of non-isothermal modeling. And he helped us work through this process where we are using the log linear primary model, incorporating that with the Bigelow secondary model and integrating it. And this will allow us to then use nonlinear regression to estimate our parameters, including the D values. And so I think about this as taking the integral of the area under that time temperature history curve and associating that with the log reduction that occurred during that time. And so here's our example plot from one of these results. This is from the Salmonella PT30 strain. And so what we're looking at here are the time temperature history. So that interval, which is what's represented on the x-axis, and then the y-axis is the log CFU per gram values. The different colors that are represented here are when we heated the sample to different target temperatures, so to 65, 70, and 75 degrees Celsius. And so from this curve, we can then determine the D values. And so here you can see highlighted by the red box here, the D values for our different strains that are shown on the leftmost part of the table. And you can see that they range anywhere from 2.8 to 4.6 minutes. And it's important to point out too, our surrogate here, the EPCM, so its t-value is 3.3 minutes, so definitely in range of the other strains that are shown here, meaning that it can be an acceptable surrogate. And so the next steps, what we're doing with this project. So we're looking at scale-up experiments and validation. As I mentioned, EPCM is an acceptable surrogate. And then we also need to, again, go through those studies where we're evaluating flour quality and baking functionality. So as we increase the amount of grain that we're treating, there's potential for that, again, to have that longer come-up time and greater transfer of heat through that load. So it may take a longer time that it needs to be processed. And so we'll have to make sure that that does not impact our baking functionality. One of the other things we noticed is that because we're applying steam under vacuum, that does slightly increase the moisture content of the grain. And so one of the things we want to do in the future was assess the potential for that moisture applied during the vacuum steam pasteurization to potentially accelerate the tempering process. So we saw about a 2% increase in moisture. One of the things we would need to do to see if this could be effective is determine if that moisture is actually absorbed by the bran layer or it's just sitting on the surface of the grain. And so to conclude here, so some of the things that I think are important for considering when we're selecting a control measure, right? So I've shown you that there are some likely options for reducing pathogens on wheat grain. But we do need to consider some of these other factors, right? So flour is a fairly low cost item, so cost of the treatment is really important. And it may be that a low cost tempering treatment, if it's shown to be effective, would be one of the most cost effective options. It's also important to consider that not necessarily all wheat needs to be treated, right? So flour that goes for industrial use, so like to commercial bakeries, we don't need to worry about processing treatments during milling because it has a kill step during that baking process. So it's really the flour that's intended for direct consumer use that is of highest concern. And one of the things I think is really critical for us to understand is what is an effective log reduction, right? So typically we think about 5 log reductions, but if the level of contamination on the grain is fairly low, it may be that a 2 or 3 log reduction is sufficient. And so to really get a handle on this, we need data on contamination levels in grain when it enters the mills. And this will help us understand what is that effective log reduction, what should we be targeting for these types of treatments. So with that, I'll conclude. I just want to take a minute to acknowledge all of the wonderful people in my lab who've contributed to this work, especially Yahweh Lin, who did the majority of what I talked about today. I'd also like to acknowledge our collaborators at MSU and the Marks Lab who helped us with our vacuum steam pasteurization development and our water activity and collaboration. And Dr. Shania Simsek, who's done all of the quality work that I presented today. Our work has been funded by North Dakota Wheat Commission as well as by the USDA. And I'd like to thank you all for your attention, and I'd be glad to answer any questions that you might have.
Thank you so much, Teresa. This is a very, very interesting and very excellent work. In the audience, from the audience, feel free to ask any questions you may have. You can unmute yourself, or you can type in the chat box. Either way would be fine. Yeah, go ahead. Okay, I probably can kick off the Q&A session.
Sure.
I got a couple of interesting observations from your slide. First one is you showed a table of survey on wheat flour. Is a 0.4% stack, 1.2% salmonella, but 0% listeria. That's interesting. That's very.
It is really interesting, yeah. Because my assumption would have been that there would have been even more listeria, right? Because we can find it easily in soil and in the farm environment. So I did think that was pretty surprising.
Yeah.
Why is that? I don't know. It made me wonder more about desiccation tolerance of listeria. Maybe it's not quite as desiccation tolerant as Aztec or something.
Yeah. Yeah, that's interesting observation, but also kind of partially explained because for many of the fresh produce outbreaks, they are commonly associated with staph and salmonella, but rarely with listeria. So there must be something going on. Listeria has a hard time to kind of survive or to get onto this these plants for some reason. I have no idea, but it's very interesting observation. Yeah, another interesting thing is the 01577 strain you used is kind of special to me.
Yes.
It seems like this strain is more resistant to chlorine treatment as well as to thermal treatment compared to the salmonella strains.
Yeah. So, okay, one thing I should clarify though. So the 0157 strain we use for the thermal studies is different. So we use the one at that point we had been able to obtain the isolate from the soy nut butter outbreak. So they're not the same strains from the two different studies. But the chlorine one, that's the spinach outbreak strain, right? And all I could think of was that it had already been exposed to chlorine, like during the spinach washing and stuff like that.
Yeah.
So I don't know if that has anything to do with it, but it was striking.
Yeah. Do you have the access to a large collection of stack strains at Michigan State?
Well, so we have the Aztec Center that's on Widow started, and we still have access to that. So that would definitely be. It's definitely worth it to survey more than one strain, right?
Right.
I know it's hard to draw any conclusions about how bacteria behave as a whole just from a single strain, right?
Yeah. I understand.
Yeah.
For the one thing I found very interesting is you included the quality test after the treatment, the bakeries, the bread baking quality. I think that's fantastic. In my proposals, I never thought of after some kind of treatment on food, I never thought of have a follow-up kind of quality evaluation products.
I really appreciate that. I have to give all the credit to my collaborator for that, right? But when we were talking about these kinds of things, she was insistent that whatever we tried, we had to make sure it would still be good quality bread, because her point, which I think is a very excellent point, no one will ever use this treatment if you don't have good quality product, right? And I guess this is just one of those types of products where that end quality can be really impacted by the treatment, right? So if you think about like produce washing, right? Ideally you would increase the shelf life, right? That's one of the things you would look for, but you don't really use like lettuce and other products, right? To make other products.
Yeah. Did you do any kind of sensory evaluations on the baker product?
Yeah, so this is still an ongoing project. We've got another year left on this, and it is our intention to do some sensory evaluation. So Shani's group moved to Purdue, and so we've had a lot of transitions, and so that's kind of taken us a little bit longer to get settled and get started on that part, but it's definitely something we're planning to do.
Yeah. I noticed your grant started in 2020.
Yeah.
Yeah, I had.
That was when I moved. Yeah.
Yeah, it's a tough time to start a project during the pandemic. So
yes, it has been. Oh, I see. There's a question here. Okay, so the question is about if we considered any synergistic treatments, so using maybe lower temperature as well as chlorine. Okay, so that's a really good question. And so at this point, we haven't considered using both like chlorine tempering and vacuum steam pasteurization, mainly because whatever solutions we want to propose to industry, we want them to be as cost effective as possible. And so honestly, I think moving forward more with tempering treatment evaluation is going to be the most important thing, because it's fairly inexpensive compared to, thinking about adding a whole nother very large piece of equipment in your processing operations. So I think your point is really good, right? That we could reduce, still make multiple hurdles, right? By reducing the levels of each of them. I'm just not sure it's most effective for this particular project.
yeah, I completely agree with that.
Yeah. There's so many things we think sound really great in the lab, but then you take them to the industry and they're like,
That's very true.
Not going to work for us. Yeah.
Very true. Any additional questions?
Yeah, I may have a quick question. So in the chlorine tempering study, I noticed that you used all the rifampicin resistant strains, but I was wondering, do you think they will respond to the chlorine treatment differently compared to the wild type strain?
That's a very good question. We have not evaluated that yet, although that is something we would want to include in our future studies when we're evaluating all the different tempering treatments. Based on, so the rifampicin resistance is generated just by point mutations, so that's why we're able to just select for those strains in the lab. Based on how rifampicin functions, I don't think there's any strong reason for us to think that it would impact chlorine tolerance, but it's still good to check that. So that is good.
Okay, because in our lab we use a lot of listeria for we generate a lot of rifampicin resistant listeria, but not for the salmonella yet. So I was just curious.
Yeah. It's fairly easy to do for all of those strains, like salmonella or Aztec. Yeah.
Right.
And we did look at growth. We're just simply comparing like basic growth characteristics, and there wasn't anything different at that point. We did not look at chlorine tolerance. Yeah.
Okay, thank you.
Yeah.
Okay. If there's no more questions, I would like to thank you, Teresa, again so much for an excellent talk and very interesting research. I look forward to some opportunities to collaborate with you.
I know, we're so close now. Yeah,
absolutely. I actually went to Michigan State in the summer a couple times for my science tournament. Yeah, so
you'll have to let me know next time you're around.
Okay.
All right. Thank you so much. I appreciate the opportunity. It's been really great. I appreciate all your questions too. It's always good things to think about. So thank you.
Thank you, Teresa.
Right.
Okay, thank you everyone for joining us today. That concludes today's seminar. I look forward to our next seminar next Thursday. Hope everyone can join again. Okay, bye everyone.
Thank you.